Ariella. It's a whole complicated thing. But chat, welcome on in. Welcome. Let's get into some science. First of all, there's a really cool graphic on this. But yeah, I like to know everything that is going on with this project. It's called the Human Pangenome Reference Consortium that we are talking about. What does this mean? There are a giant series of words in here. There's also this hilarious graphic. I think it's supposed to be a fish. A stylized fish is a form of DNA. I do want to point out what this is because it's on a lot of their studies. This is a chromosome in a helical loop. It also has an extra loop inside of the chromosome that it's a usual 3D structure that chromosomes do have and are like readily we know the ultrastructure of DNA has all this looping mechanisms and it allows it to compact a little bit more. Hi, Jay Brains. Welcome in, Jay. So that's just why I wanted to point it out. I don't know if, again, they're going for a fish look or not. But what exactly is this? So the Human Pangenome Reference Consortium. The project is funded by the NIH, so that's the National Human Genome Research Institute, to sequence... Jay, thank you for the bitties, Jay. And assemble genomes from individuals from diverse populations in order to better represent genomic landscapes of diverse human populations. Tomato paste, not ketchup, a half cup 2011. It got mocked due to the amount of... Okay, gotcha. Thank you, Alex. So there was something about the tomatoes. Very cool, but also imagine 20 years from now on your hologram saying, wow, I remember when we said 47... You're not wrong, Ariella. You're not wrong, but remember, for the longest time, we're going to go into historical elements of this. The reason this is interesting is because the Human Genome Project, where when we said our genomes were sequenced, that was age 1 genome, and that they were basing further information on, and disease states, and treatments, and just like your fundamental genetics and comparisons and evolution and everything, based on a single genome. They're now doing new kinds of sequencing, so we're going to go into the sequencing methodologies of this. And they already have 47 sequences of 47 genomes, 47 different people, and they're increasing that number to 350 by the middle of next year. So the idea is that this will give us much, much more visibility and range to actually analyzing what the genome means, what the genes are within the genome, how they correspond to disease states, to differences between humans, and much, much more. But let's get into some of the science behind how this works. So there's actually a really cool intro video on like what this original goal was. So they released this 2 years ago, this video, as what the goal of the project was. And I always think it's kind of neat to see what project goals are when they start, and then we'll go to historical element and talk about how they actually did the first human genome, and then we'll get into some of the importance behind the study.
More than 20 years ago now, my team at UCSC posted the first human genome sequence on the internet.
UC Santa Cruz, and by the way, all their buildings are that kind of brick. Not that it especially matters, it was just something that when I was in California at the UC school system, like, huh, they are all this weird gray brick. And I thought that was kind of interesting. Guys, also, as we said, my shirt has the lovely Moco Locos logo on it. Guys, go check out the lovely, amazing Moco Man. First multicellular genome sequence was the nematode, indeed. And that is a very cute emote, too. Ariella. See, I like this logo much better than their little weird fish. Because it's representing different genomes of different people and also the tree of life.
We went on to create the UCSC Genome Browser, essentially a web-based digital microscope that is now...
And this genome browser is used everywhere. So this is a tool that allows you to look at the different genetic components and like the broad ranging genome and seeing like how they align to other animals. So if you want to see, like, here is a genetic sequence of a mouse, let's say, and you want to see how it connects to a human, you can line these things up now in this amazing tool. Risto and Looking Cool Zedman, welcome on in. How are y'all doing today, Risto? How was the rest of your day, my friend? I hope you got some lovely sleep last night and that you got some good working. And Risto's been pushing himself too hard, chat. He's, I don't like it, Risto, when you push yourself that hard. I hope you're doing well. And Zedman, how are you doing, sir? I hope you're doing well. I hope you're getting ready to put on that CPAP and feel better, sir. All right, damn, so tired. I'm probably going to try a nap. Wait, Risto, it's too late for a nap. You gotta be getting into bed soon, Risto. Group photo also made me think, have you seen this? Oh, yes, Alex. I actually have a photo like that as well, Alex, in front of that building. There's a lot of COVID variants. Yeah, they're all these COVID variants, all these genomes. You can line up the different COVID variants and see how they work. Like what similarities their differences are between them. Like, so if these are all different amino acids, you can line them up and see if there are sequence changes or amino acid changes. It's a very powerful tool. Getting the house ready for my mom to visit tomorrow. Very nice. Zeddy, you're gonna try to get some sleep though, right, Zedman? Zeddy, folks, is working on trying to play the new Tears for Fears Zelda game. I know it's not Tears for Fears, but that's what we're gonna call it. And he's on everyone's channel who's playing it, and so he's just gonna get the whole story spoiled for him. Just terrible. Rista, how are you feeling about your event, my friend? Are you guys? Ristarok tonight is having a big event on his stream at midnight. Midnight Pacific Standard Time. East Coast is 3 a.m. He's part of a mental health raid train, talking about the importance of mental health and loving thyself, amongst many more. He also plays games. He's also got a shirtless calendar that he sells to benefit a nearby orphanage. Go check out Ristarok. Hi, Paw Prince. How you doing, Paw Prince? You're avoiding spoilers. Zeddy, listen, Zedman, I have to ask you, sir. Everyone here is so excited about this new Zelda game. I saw one person going off about how it's only 20 frames per second and how they just cannot, at all take the frames per second rate and that it's a rip-off of the last game. That's what I've heard, Zedman. That's what I've heard. I've watched people play it. It looks very nice. The graphics I'm not enamored with, but I'm just imagining if you could do that with Witcher level graphics, how cool it would be. But it does look like a fun game. Even you can cook in this game, chat. You can cook. Wellness and coping skills tonight. Everyone talking about the illness side of things. We're going to go with the wellness side. Risto, it's easy to go dark. It's hard to go light. Hello, Epic of Sam and Blueberry.
Hello there.
It's hard to go light, Risto. And so that positivity, I think, would be very beneficial for the party people. Moonrise, how are you liking the game? I hope you're enjoying it. All right. We're also going to give a shout out to Lovely Looking Cool Zedman as well. Zeddy, are you still streaming on Mother's Day, sir? Are you still streaming on Mother's Day? Making sure Zedman's mom is coming over tomorrow. Are you loving it so far? Moonrise, I have the last game. I have not finished it yet, and so I figured no getting the new one until I actually go and finish it. Loki, the series was missing something. Okay. Doing Mother's Day tomorrow and Sunday. So very cool, Zeddy. We're doing Mother's Day on Sunday, and then we're doing science art stream tomorrow instead. Last Zelda game was the first one. Oh, gotcha, Moonrise. I played some of the older ones. Hi, Supernatural. Welcome in. Let's see. I actually just got home from taking Grandma Risto and her bestie to dinner for her. Aww. That's beautiful, Risto. Why are you so beautiful, sir? Why are you so beautiful? Owen Wilson saying, wow. Yes, Hugan. Hugan. Yes. Yes. I've heard also that Link sounds like Owen Wilson. Hugan. Is this true? Guys, also go check out the lovely and amazing Looking Cool Zedman. The coolest looking Zedman of them all, folks. Full-time variety content creator extraordinaire. Go check out Zeddy. She's the best grandmother who deserves all the things. Risto, I will fight you. Not really. I will suggest, Arista, that my grandmother is also pretty amazing. But I do like the fact, Arista. Hi, Crystal. Welcome on in. Sweet and Bell, thank you. I do like, Risto, that you fight for the love of your grandmother and the fight that she's so amazing. That brings love to my heart, Risto. Love you. All right. So, for those coming on in, we're talking about this human pan-genome project, meaning that they have gone from having just one human genome that we're comparing everything against to now multiple, which is a huge jump forward in the landscape of science. 81 is still up with her lawn. That sounds like my grandmother, Risto. She is 83, and right now is at home by herself because my mom, dad, and my brother are in Spain. And so I'm calling her every day to check in on her, make sure she's all right. Miss Kathy, the lady who like helped take care of us, she still goes every day to help her around the house and whatnot. And Risto, instead of actually embracing that help, she just tells her to go home. So like she'll come in and check on her, be like, all right, go home. And then she's like, oh yeah, today I trimmed this like giant tree. I'm like, what are you doing? You fall. You're like, don't do this. Why are you doing this? It's a whole thing. It's a whole thing, Risto. Still fought the neighbors about it. She still walks 2 miles around the island every day.
Don't you like me a little bit?
Ew, I don't even like you a little
I love
bit.
it. Hi, Bits
I heard you
Gaming.
like me a little bit.
Well, maybe just a little bit.
Grandma, mow your lawn in heaven. What the heck? Oh, Alex. Hi, Bits Gaming. Master Lee, how was your dinner tonight? I hope it was delicious. Handsome scientist, you're right. Zedman is here. Zedman is here. He's very handsome. He's very handsome. Guys, if you check out Big Gaming, go check out Big Game. Legendary content creator extraordinaire. He was doing a lot of
SciAnts
game
know you.
dev
You're the
today.
cutest to know that you deserve it, Juno. You a star over here.
Lot of game dev
First,
today.
hard like Uno.
The lovely Big Gaming. Working on Homeward Hound today. And also, as a baby, Bit Gaming. I don't think we even
Where's
finished.
my drink?
We didn't finish all the
drink?
redeems, Bit Gaming. Thank you, Ariella. Oh. This is lovely, Ariel. And pan cook it and see what happens. Doesn't seem very scientific. Hugan, you're very silly, my friend. We will get into why it was called Pangea. They were still going after Raid. That doesn't count. That does not count. Is there? I have no idea what a Koopa is, Epic of Sam. It is a frog. My uncle broke a couple of ribs falling off his roof when he was trying to sweep the snow off it. He was in his 80s. Recovered fine, but there's a trifecta of tempting fate. Moonrise. Luckily, my grandma's down in Georgia, so there was no snow. She moved from Hungary, though, which did have snow. Koopa turtles from Mars. No. It is just a little, it is a frog on a lily pad. Tea strainer, if you will, Epic of Sam. Although, the Koopa would be very cool, Epic of Sam.
Now been used by a whole generation to explore life's code. The International Human Genome Project was a monumental effort. UCSC played a small but pivotal role. In that project, we became the first species to read our own DNA script. Since then, those 3 billion A's, C's, T's, and G's have become the basis for the study of human biology and genomic medicine.
So 70% of the first genome came from one individual. So you can imagine if we're just having, if there's a genome out there that we're comparing everything against and most of it is from one person, a lot of things are going to look weird because you don't know what normal actually is, right? You don't have a range of normal. All you have is this one thing to compare against, and if it's any different, you don't know what the consequence may or may not be. Debs, I cannot. Debs, you even spell Brazil differently. Chad, Deb spells Brazil with a Z, not an S. I never even know what to do with that, Debs. I was like, what is this? It's fancy, Debs. It's fancy.
Studies estimate that as much as 10% is still missing.
We've known for decades that humans share 99.9% of genetic material in common. We are amazingly similar. We've realized that 1%.
And then Alex says natural mutation would not be considered within a small sample. No, exactly. So, Alex, we just wouldn't know what variance is variance of meaning and what variance is variance of, like, actual biological meaning. The two things we'd have no idea on.
Since DNA is really inadequate as a representation of human diversity. Human genomes are so big that 0.1% is actually millions of differences. Understanding these differences is helping scientists predict health risks and gives us real insight into disease.
Sleep well, Risto! Risto, good luck tonight. Good luck tonight, Risto. If I'm not there, it's because I'm sleeping. And that's not a commentary on me not wanting to be there to support. It's just, I sometimes get a sleepy, Risto. I get the sleepy. Love you, Risto.
One genome simply cannot represent all of humanity. The Human Pan-Genome Reference will be a key step forward for biomedical research and for personalized medicine.
I study mutations associated with cancer. Having a better genome reference will make it easier to find these cancer-specific mutations and make sense of them. Diversifying the reference genome increases our knowledge and could lead us to greater discovery.
So what they don't talk about openly is that the original genome that was sequenced actually had gaps in it. There are regions of chromatin that are very difficult to sequence because they're very, very tightly compacted. And because they're so tightly compacted, it's tough to get at the sequence reads. And so newer technologies allow us to actually get there and to fill in those extra gaps. So even that, total human genome sequence from 2003 was not the full sequence. And guys, I love your face, Professor Canuck. Please go follow Professor Canuck. Background music kind of started sounding like the Halloween theme. It is from the UC Santa Cruz school system, Professor Canuck. So you never know how silly willy they look like. Always, Professor Canuck. I apologize if it was late. I can't remember if we already did one or we didn't do one, so I just want to make sure we get another one in there. And, if we do 47 of them, it's not a big issue. We can just keep going on to the sequencing. Crocheting. Gotcha. Thank you, Squalls, I know now the difference between crocheting and knitting in terms of the tools, but I do not know when looking at it, like the product where it's from. I'm sorry I'm not getting any sleep tonight. I don't know why you're sorry, Blueberry. And yes, Big Gamey, you are correct, Big Gamey. Chat! Let's continue.
Our job is to build that representation and make it easy to use. It's a huge challenge, but it's really important. If you rely on one genome for all your comparisons, you're blind to many types of variation.
So this is why this study is so massive, that they did all these additional genome sequencing experiments to figure out more what they can compare against. I'm sorry because I should be sleeping, but not sleeping. Okay, Blueberry, that's not all me. You don't have. I am not in charge of your sleep schedule, Blueberry. You don't have to say sorry for that. Yeah, it's okay, Blueberry. It's okay. It's okay. Blueberry, it's fine! Big Game is here for you. So this is why this is such a big deal. Getting more sequences of more individuals from different and diverse populations will allow for experiments to be better and for be more informative when studying humans and making informative medical decisions. And so what they're saying too is that the quality of the genome is going to be better, meaning that they are having a better representation of the genetics in terms of the sequence, and they're not having as many gaps as the original genome sequencing did. So they're getting the entirety of the genome of many individuals that they'll be able to hopefully have much better information against.
These repeat-rich sequences were once thought to be unsolvable, but recently we've made leaps and bounds. With nanopore sequencing, we can get ultra-long reads of hundreds of thousands of base pairs. These reads can span an entire repeat region. Some of the regions where there are gaps in the human reference are actually among the richest for variation.
What it means if there are repeat elements in a genome, it means that if you look if you're walking down the genome, it's the same sequence over and over and over again, and it is very difficult to sequence that region, because if you're trying to sequence it, if you get a segment of it and you're trying to match up where that segment goes, you will not know where it goes because everything's repetitive. The only way to know exactly where everything fits is if you can do that entire sequence in one read, in one piece of data collected during the genome sequence. And that's really difficult. Most technology requires really short sequence reads in order to identify what they are. You get the sequence read and then you have to match it up against a reference genome. Again, though, if you're getting these repeats, you don't know if that's the beginning of the repeat segment, in the middle, in the end, or elsewhere, anywhere in between. So getting these new types of sequencing technologies will really help us actually figure out what's in the genome.
We're finding a lot of information that could be important to understanding human biology and disease.
And they're trying to have, and this is kind of neat that they point out, a way to visualize such a genome. So because there's so many, like they're doing an additional 47 genomes, right? But they're going to do a total of additional 350. And the question is, well, how do we visualize all these differences? If there's so many genomes all at once, how do I know what I'm looking at? And how do I know if it's a worthwhile thing to look at? And so that's one thing they're going to be trying to do, is building a tool for other scientists and other individuals to look at the genetic information and make comparisons and get information from it that they otherwise wouldn't know how to do. And so this is an example of the visualization tool. So this up on the top is the region of chromatin that you're looking at, and then these individual lines of different color are different individuals, and the length that the color goes shows the length of that genetic sequence that they have in there. So the block is the genetic sequence that you're interested in, and you can see some individuals have the entire block, others have very different sections of the block, and then if that block is important to some kind of phenotype or effect, you might be able to figure out this way, which I think is a really, really cool tool to have that. Pemarley wants to thank you, but type Pemarley, help! Hi, Pemarley, aka NMD96. So,
Genomic sequence.
Let's actually get into what is genomic sequencing, because they kept talking about it in this video. That's the whole crux of this paper. Let's try to figure out what exactly is genome sequencing.
Sequencing is a process for analyzing a sample of DNA taken from your blood. In the lab, technicians extract DNA and prepare it for sequencing. Within every normal cell are 23 pairs of chromosomes.
For humans, although you can do these kinds of sequencing experiments right in any kind of animal, which is actually going to be the second story that we talk about tonight.
Chromosomes are structures that house DNA. DNA is coiled into a shape called the double helix. The double helix can be unwound into a ladder shape. The ladder is made up of paired chemical letters called bases. Altogether, our DNA contains about 6 billion such paired chemical bases. There are 4 bases in the DNA alphabet: A, T, C, and G.
And remember, that's how they pair. A always goes to T. Oh, Tarzan. You're Gucci, Tarzan. Tarzan, in order to do it, you do poke shop, poke ball. So something like that. And Tarzan, the number at the end indicates how many Pokeballs you would like to purchase. If Tarzan, you ever need any help, all you do is you type poke help, and that brings you up all the commands. Buttspot, no. Naughty Buttspot. Naughty Buttspot. And yeah, Squalls, it's a ton of data.
In DNA, base A joins only with base T, and base G joins only with base C. To read the sequence of bases in DNA, samples are inserted into a sequencing instrument, where high-frequency sound waves break the DNA into smaller pieces.
Hi, Astronomy Show. Astronomy Show, I will say, I don't think it had an I in there, but it is a little Gattaca. Astronomy Show, how are you doing? Welcome the heck in, friendo. Guys, check out the lovely and amazing Astronomy Show, who does, in fact, as their name suggests, astronomy. How you doing, Astronomy Show? Welcome in. Indeed, NMD. NMD, remember, that's the movie that we watched for movie night where I thought it was going to have, like, battles and flying and shooting ships and stuff. It didn't have any of that. NMD. Had none of that.
That are only about 600 bases long. Special tags.
So this is what they're saying. We have to, you have to chop up the genome into smaller bits that the machine can read. And that's the enzymes and the mixtures and the chemicals that you're using are optimized for these smaller reads. Zergrush, all your bases are ours. Alex, I thought it was, I thought that movie Gattaca was going to be like that movie, oh my goodness, with John Travolta. I thought it was like Battlefield Earth or something like that. I thought that was Gattaca, and so the whole movie I'm waiting for the space battles and John Travolta's alien self to show up. It was a very different movie, Alex.
Tags are added to the ends of the fragmented DNA. These tagged strands of DNA can then attach to a glass slide. In a sequencer, each piece of DNA is copied hundreds of thousands of times, which creates clusters of identical DNA fragments. Next, the sequencer reads the DNA, one base at a time, using different colored tags for each DNA base. Special sensors within the machine detect the different colored tags. This sequence of colors reveals the DNA sequence of each fragment. Powerful computers piece together these individual DNA fragments.
And what's interesting is that we just talked about you can't piece together these repetitive elements if there are repeats in the genome that look the same. If you try to piece them together against this reference genome, if you're trying to add them to it, if they are far too similar, then the computer won't know where to put them. And so that's when you need these longer sequence reads.
And reveal the sequence of your DNA. Then a team of medical experts uses specialized software to analyze and compare the DNA sequences to identify the handful of variants that may be important for your medical care. Discuss the results of the sequencing of your DNA with your doctor and a genetic counselor.
More so the genetic counselor, the regular MD is going to be like, I don't know what to do with this, because individual, just single polynucleotide differences like they showed aren't necessarily always informative. You need something to inform against, and hopefully these additional sequences will help with that. The movie surprised me for being so good. I thought it was fun, Astronomy Show. I don't remember really liking it, but I don't remember really disliking it either. Medical genetics is a residency program now. Oh, very cool. That sounds nice, Ariella. I would imagine that with personalized medicine, we're going more and more into that zone as well. So chat, let's talk a little bit more about how to sequence the human genome, and then we'll go on to how to even interpret it before we round out with another talk about the pan genome, and then we'll move on to the next study as well. There's also a cool story actually about how the human genome was sequenced that I think we might touch on as well, because that's actually a pretty neat story. The message about knitting crochet makes it maybe crafting squalls, but we'll see. My brain always isn't good with that, but we'll see crafting squalls. Thank you.
You've probably heard of the human genome, the huge collection of genes inside each and every one of your cells. You probably also know that we've sequenced the human genome. But what does that actually mean? How do you sequence someone's genome? Let's back up a bit. What is a genome? Well, a genome is all the genes, plus some extra, that make up an organism. Genes are made up of DNA, and DNA is made up of long paired strands of A's, T's, C's, and G's.
Thank you, NMD. Garden Gnome, Hugh. Last I saw, I don't think we had Nomio to deal with, but I'm not entirely sure. Not entirely sure, Hugh, about the garden. No, but maybe Nomio. Maybe Nomio knows.
Your genome is the code that your cells use to know how to behave. Cells interacting together make tissues. Tissues cooperating with each other make organs. Organs cooperating with each other make an organism. You. So, you are who you are in large part because of your genome. The first human genome was sequenced 10 years ago and was no easy task. It took 2 decades to complete, required the effort of hundreds of scientists across dozens of countries, and cost over $3 billion. But someday, very soon, it will be possible to know the sequence of letters that make up your own personal genome, all in a matter of minutes.
So I will say this video is almost 10 years old. The science behind it is still correct, but even 10 years later, we're just now getting to sequencing, like for that reference genome project, 350 different individuals. Right now, it's not so much the price, although the price is, I think, right now, instead of being billions, Tarzan, it's a few thousand for sequencing a genome of your organism of interest. Like, if you want to go out into the world and be like, I want to know what the genome of this thing is that hasn't been sequenced, it's probably a few thousand, which, relatively speaking, is really nothing in the grand scheme of everything. They don't yet have it. The technology isn't minutes yet. It's usually overnight, so also getting faster. But it's still relevant today.
And for less than the cost of a pretty nice birthday present. How is that possible? Let's take a closer look. Knowing the sequence of the billions of letters that make up your genome is the goal of genome sequencing. A genome is both really, really big.
Dame Karen, I do have a video on it. If you'd like, we can watch it after this, because actually, it is a fascinating video, Dame Karen, of 2 competing groups fighting against each other and each trying to come out in the end the successful one who's identified what the data actually is. It feels like it's coming out of some novel, if anything. We'll watch that next, Dame Karen. So, I mean, considering we have better look at a woolly mammoth genome than ourselves, it's still mind-boggling. And Tarzan, even in the woolly mammoth genome, there's still these large gaps. And those large gaps are, again, in part because we just don't have a good technology to sequence it. Because for a long time, the proteins and enzymes and molecules that we put into the sequencer are dependent on these short reads. And making long reads is just... because what you need to do for a long sequence is you have to have 2 adapters attached to either end, and then your enzymes connect on those adapters and go opposite, and they sequence the whole strand. In the past, Tarzan, the tools that we have, those enzymes that are running along the sequence and replicating it, they cannot sequence that long of a sequence, like 600 bases or more. And so finally, they've had to adapt those old pieces of tech to be better and better for us to be able to get that kind of information. Dilly-the-do! Hi, Dilly-the-do. Dilly-the-do, how are you doing today, guys? If you're not checking out Legendary Dilly-the-do, please go check out Dilly-the-do. How you doing, Dilly? How you doing, Dilly-the-do? Tonight we're doing SciAnts News of the Week. And there's a really, really cool breakthrough study on human genetic sequencing that they've sequenced 47 additional people to look at their whole genomes. They're aiming for another 350. And so the idea is that you can do comparative genetics between people, and they're from diverse populations as well, to figure out, okay, a mutation here yields a disease state. A mutation there doesn't yield anything across most populations, versus maybe in this population is something, but it's because of another mutation elsewhere in the genome. So you can start making those really advanced studies looking at relationship between genetics. So that's the hope, anyway.
And very, very small. The individual letters of DNA.
Are they planning on using AI to do the sequencing and fillers? So the AI Coffee Rocks, there are machine learning programs that they actually used to predict what a gene's function might be, but they don't use it to do the sequencing itself. So the AI isn't good enough because we need the actual sequence to be able to make any inferences on what the genes and how they're working on, like, and what is normal. If we have AI start filling that in, that could throw off the whole system. What they do use it for is if you get a genome sequence that you can tell, okay, this sequence is a gene. There are machine learning programs out there. One is called Pilgrim, another one is called Imp, another one is called Tribe. They have these unique names to it, and they are gene prediction tools. You put in your sequence of your unknown, and it actually uses known sequences and known structures to predict what your mystery gene might do. At worst, coffee, it might tell you there's regions in this gene that make this kind of protein, and there's other regions in this gene that make a different kind of protein, and together maybe it makes something like this. But it's really cool how they're using it on the AI front, but not so much yet on the sequencing. We need the actual, like, the hard information on that front. We can't make any guesses on it. So this is how they deleted the chromosome X deletion. So Tarzan, for chromosomal deletions, there is a process called karyotyping that they have used in the past, and it's actually something that you can do even at home. I don't know if you can do it on your own cells, without extra tools. I know you can do it on a fruit fly one, but this is an example of that, Tarzan. So this would be a human karyotype, and you see eventually you're getting a map of all the information here. And so for missing X's and X deletions, you just be missing an X chromosome here. So instead of XX, let's say you just have one X. This also will show Fragile X, Tarzan, where in Fragile X syndrome, part of the X chromosome is broken, and it's connected by a really, really thin strand. You can see that on a karyotype as well. You can see very large gaps in this, but you can't see anything at really tight resolutions. Why is the genome sequencing important? What's the goal of using this information? So Squalls, let's say that you're really sick on something, and we have no idea how to fix it. Squalls, what we'd be able to do is to take, if we sequenced you, we can compare it against the genetic sequence of these known sequences and find out, okay, what is normal and what is abnormal, and hopefully identify a way for you to get a personalized treatment to make sure that you get better. So this is a way to predict the genetic reasons of disease and then maybe identifying treatments for them. Without this information, there'd be no way of doing that, right? Because Squalls, if we didn't have this reference genome, if I just sequenced your genes, your whole genome, if I have nothing to compare it to, I have no idea if it's correct or not, right? Or like what's wrong. All I would know is, yes, your genome is there, but I'd have, again, no way of knowing what to do and how to change, how to fix it. So Squalls, that would be the big reason why. And the reason they're doing 350 people is they're getting a bunch of different populations. So let's say that you're Eastern European. If I were to get a bunch of Californians' genome sequence and compare it against yours, we would find differences, Squalls, that are probably not a result of, that are not causing a disease state. They're just differences because you have two very different populations. So if you have more of these reference genomes from different populations across the world, you can figure out what are just genetic differences because you're living in different places and you've, interacting with different people versus which ones are the things that can actually contribute to disease. So that's the big goal behind it. Does that make sense, Squalls? Ariel says, caution, sometimes you get pretty big surprise when you do this. Yes, Ariel is right. So Tarzan, they used to do these kinds of karyotypes in the classrooms as well, and that can lead to different effects. Like sometimes you can have an XXY or an XYY without being able to tell there's anything off, and then if you get it in class, that would be very uncomfortable, right? We went because my son got checked for Fragile X because we're both autistic, and it's common to find in autistics apparently. I can let Tarzan, let me show you what it looks like on a karyotype. So this is like a nice... Hi, Chris McBricker. Welcome in. So this right here is that little indent, Tarzan, that you'd be looking for on Fragile X. So there's that little... It's that almost a string connecting the strands on like the tips of the X versus the body of the X. And so that's a little bit of like what Fragile X looks like. I think if we can blow this image up. So right here, Tarzan, that little extra bleb is right here. That's the Fragile X component there. Right there. You can also find out your XY. Androgen sensitivity if you don't want to have the kit on Tuesday with no warning. So Ariel, it's yes, you're right. So crafting squalls, that is a effect of having a different kinds of sex chromosomes than you might be expecting. Squalls, how do we determine the bases? How do they pick a perfect one as compared? So crafting, you wouldn't just do a single genome, you'd get an aggregate average answer. So if you see across your 350 individuals, 80% of the time, this gene is this particular length and has these particular bases. So it usually has an A, T, C, G, A, T, C at that particular site. Then that's probably the average answer of it. And you might have differences in there that don't make a difference, but there might be some in there that do make a difference. So you're not just comparing against a singular person, but rather the average or the aggregate. Hopefully that makes sense, Qualls. Which is why I was concerned about AI. If AI, you could control it, meaning it would be possible that treatments prescribed would actually make the subject worse. Exactly, Coffee Rocks. The hope is that there would be no influence of AI on that sequencing front. Because you're right, Coffee. If we're inputting AI, you wouldn't get the raw answer. You'd be getting a predicted answer, and like you're saying, Coffee, that could be a potential negative for the patient. So you'd want to get close to the answer, or as close to the answer as possible. Yeah. Awesome. Thank you, Squalls. I'm glad all this. And Ariella, thank you so much, Ariella. Okay. So wait, how would you get your genome sequencing done? There are companies, Squalls, that will do this for you. It's honestly the biggest issue right now isn't getting the sequencing done. It's more getting the analysis done. So there's, and actually, Squalls, if you do get your genome sequenced, there is still the technology to make it so there's no gaps. My understanding is the common, like, one that you can purchase still has some gaps in the reads. And so to get the no gaps is either more expensive or it's not fully yet available to everyone. But yeah, there's companies online will do it. The hard part is actually reading the information and knowing what it means. Because Squalls, the data you would get back is a file, like an Excel file, with just bases, A, T, C, G, for 6 billion. And that's your genetic sequence, right? So that's not going to tell you anything. You have to have someone to be able to read it to you, which means you have to have the training either in the computer software to analyze it, to compare it against an existing genome and to read it, or like a genetic counselor.
The A's, T's, G's, and C's are only 8 or 10 atoms wide, and they're all packed together into a clump, like a ball of yarn.
Hello, Kennedy. Welcome in, Kennedy. How you doing, Kennedy?
So, to get all that information out of that tiny space, scientists first have to break the long string of DNA down into smaller pieces.
And they're trying to avoid doing this now, so that you'd be better able to sequence those really difficult sections of the genome. Curious if there's anything abnormal. So, Squalls, your first bet would be, they don't do this anymore the way they used to. There's a company called 23andMe, and what they do is you send in a saliva sample, and they give you, it's a kit they send to you. You spit into a tube and you send it back, and then you go online and you read out your results, and it'll tell you where you're related from, like from their database, where are you most closely related to, like what percentage of X, Y, and Z you are. They used to have, Squalls, an option there that would tell you disease risks. They have done away with that because they got sued. They used to be able to, like, predictive mutations that could be negative against your health, like I think muscular dystrophy. I think there was a couple of other ones too. They got sued because it's not actually definitive. It's just suggestive that you could get those diseases, and they got sued against it due to misleading components. So Squalls, right now, I think for your immediate need, probably some of those lower tier sequencing runs or packages would be more beneficial than a whole genome, because the whole genome, again, it's not the price of buying it, it's the price of having someone read it and analyze it. That would be the issue. It would unnecessarily work. Well, it could unnecessarily. So Dame Karen, when I had mine done, they still had it, and they covered up that region, so you had to click on it to open it up. I never clicked on it, and I do not know. I haven't gone into it in so long, Dame Karen. I don't know if they actually retroactively got rid of it, but I would be curious to figure out if they did. The end discussion last week at the CrimeCon Writers Conference, and I've done an intro to genealogy presentations. Nice, Kennedy. Very cool.
Each of these pieces is then separated in space and sequenced individually. But how? It's helpful to remember that DNA binds to other DNA if the sequences are the exact opposite of each other. A's bind to T's, and T's bind to A's. G's bind to C's, and C's to G's. If the ATGC sequence of two pieces of DNA are exact opposites, they stick together. Because the genome pieces are so very small, we need some way to increase the signal we can detect from each of the individual letters. In the most common method, scientists use enzymes to make thousands of copies of each genome piece. So we now have thousands of replicas of each of the genome pieces, all with the same sequence of A's, T's, G's, and C's. But we have...
Internet cut out for a sec. Sorry, it's repetitive. These DNA companies can't just willingly give your diagnostic information. Yep, exactly. Even if you did, though, you're a scientist who'd understand. A lot of people don't understand. No, Dame Karen, no, ma'am. I would also panic. I would 100% also panic. If I'm like, oh, I'm gonna get my... Even, like, Dame Karen, it's like with the kiddo, right? As soon as there's the mention of something wrong, even though rationally I know doesn't make sense, the thought of it would be a little much, you know? And so I would 100% get on that panic front too. What would happen if the letters connected that aren't supposed to? They wouldn't connect, Squalls. So A always goes to T, C always goes to G. If they don't connect, there's a gap, and then the information can't happen. They will never add in the wrong base pair. What you might happen is, during your replicating your DNA inside your cells, there might be an error, and your DNA machinery might remove one of the base pairs and put in a different one, and inducing a mutation. But beyond that, you wouldn't have it happen. Why? So crafting Squalls, there's two sizes, and those two sizes, because of how they're linked, they'd have to connect a certain way, and otherwise, if you're putting in the wrong one, they don't connect, and it just falls apart. It's just the biochemistry of the organism. Insurance. So supposedly they're not supposed to, Alex, but yes, that is the terror of it. If an insurance company gets a hold of your genetic findings, then they can adjust your premium based on your likelihood of diseases. It is, Ariella, but yeah. Is it like a puzzle? It is, NMD. The way these fit together are kind of like a lock and key, and that's why it's always A with T, C with G, and not other kind of pairs coming together.
Have to read them all somehow. To do this, we need to make a batch of special letters, each with a distinct color. A mixture of these special colored letters and enzymes are then added to the genome we're trying to read. At each spot on the genome, one of the special letters binds to its opposite letter. So we now have a double-stranded piece of DNA with a colorful spot at each letter. Scientists then take pictures of each snippet of genome. Seeing the order of the colors allows us to read the sequence. The sequences of each of these millions of pieces of DNA are stitched together using computer programs to create a complete sequence of the entire genome.
So it's cool they're using lights to figure out what each sequence.
This isn't the only way to read the letter sequences of pieces of DNA, but it's one of the most common. Of course, just reading the letters in the genome doesn't tell us much. It's kind of like looking through a book written in a language you don't speak. You can recognize all the letters, but still have no idea what's going on.
And so, Squalls, if we sequence your genome, that's what we would have. Like, without having the decoder manual, you just have a series of 6 billion A's, T's, C's, and G's. Yes, NMD is on our partner Poosh, folks. Go check out NMD. Blueberry is also on our partner Poosh, folks. There we go.
So the next step is to decipher what the sequence means. How your genome and my genome are different. Interpreting the genes of the genome is the part scientists are still working on. While not every difference is consequential, the sum of these differences is responsible for differences in how we look, what we like, how we act, and even how likely we are to get sick or respond to specific medicines. Better understanding of how disparities between our genomes account for these differences is sure to change the way we think, not only about how doctors treat their patients, but also how we treat each other.
It's a little bit on how genetic sequencing works. Let me find this video for Dame Karen. Dame Karen was curious about the history of genetic sequencing. So let's see. Let me re-find my video here. Ha! I got it. I got it, Dame Karen. It's actually quite a spicy story of how it actually works. See, there we go. I knew Blueberry. You'd be streaming Blueberry. I might enjoy that a lot. I might enjoy that redemption a lot, chat. Okay.
Packed inside every cell in your body is a set of genetic instructions, 3.2 billion base pairs long. Deciphering these directions would be a monumental task, but could offer unprecedented insight about the human body.
Again, and so then Squalls, if we have more information will then allow us even more data to look at, right? And it'll get even more information coming in. Recessive genes show up in the genome that otherwise would not at sample-wise. So yeah, Coffee, we might not know with one genome what a recessive genes are, right, Coffee? Because if you get someone who's heterozygous, meaning there's a wild-type copy and then your mutant recessive copy in a lot of places, and so they're, phenotypically normal, you'd never know, Coffee, that other copy is a recessive mutant. You'd have no idea of delineating between the two of what's what. But with having so many folks sequenced, that's how we'd be able to identify who has the recessive copies and what those recessive copies mean. Guys, go check out the lovely and amazing Supernatural Writer as well. Workout streamer, co-working streamer, beauty streamer, food and drink streamer, Christmas tree putter upper, and so much more. Guys, go check out the amazing, breathtaking Supernatural Writer. We love you, Super! Thank you, Blueberry. I'm not worthy for that, Blueberry. I'm not worthy. It also freaks me out. Blueberry, make sure you ask before you make me mod, because I like to, if I'm a mod, I like to take it seriously, and if I'm just loosey-goosey given mod status, I get scared. Because it's good to ask. It's good to ask before you throw. Alex, yo, it's good to ask before you mod someone. If I show up to a stream where I haven't been, and all of a sudden, for a while, and all of a sudden I have a sword, I'll be very concerned. I'm like, well, I don't know where this came from.
1990, a consortium of 20 international...
Thank you, Jim Care. Okay, let's continue, chat.
research centers embarked on the world's largest biological...
It's time to say something nice about yourself.
I have amazing moderators without whom I would not be where I am today, and that also suggests that I might not be a terrible person, because if the moderators who are so kind and amazing are with me, that must mean I'm somewhat of a worthy human being. Or something other. Alex Dixon, I have you as a friend! You as a friend, Alex!
Collaboration to accomplish this mission. The Human Genome Project proposed to sequence the entire human genome over 15 years with $3 billion of public funds. Then, 7 years before its scheduled completion, a private company called Celera announced that they could accomplish the same goal in just 3 years and at a fraction of the cost.
So this is the war. This is the war that broke out, Dame Karen. Company versus academia. Hi, Mad Chef! Alex, this is not half bad.
I like your pants around your pants.
The two camps discussed a joint venture, but talks quickly fell apart as disagreements arose over legal and ethical issues of genetic property, and so the race began. Though both teams used the same technology to sequence the entire human genome, it was their strategies that made all the difference. Their paths diverged in the most critical of steps. The first one. In the Human Genome Project's approach, the genome was first divided into smaller, more manageable chunks, about 100.
And this is, again, like, this is the old tech of having everything be shortened. The new tech doesn't necessarily require everything to be this short. No, you, Blueberry. TTS's Big Gamey, my favorite. Sleep well, Debs. Sleep well, Debs. My favorite Big Gamey is when Irish John made me into a TTS character, and that clip reveal, Big Gamey, where Jern says the TTS's, and then he like shakes around and he like pulls out his S a little bit. Big Gamey, it made me chuckle so very much. It brought so much joy to my heart. You have a chef chop up your DNA? I mean, it is the enzyme, Dame Karen. Where's the clip? On the internet, Blueberry. On the internet. You have to search through Irish John Games' clips, and then you can find it. It's somewhere in Jern's clip drawer.
150,000 base pairs long that overlapped each other a little bit on both ends. Each of these fragments of DNA was inserted inside a bacterial artificial chromosome, where they were cloned and fingerprinted. The fingerprints showed scientists where the fragments overlapped without knowing the actual sequence. Using the overlapping bits as a guide, the researchers marked each fragment's place in the genome to create a contiguous map.
And like we talked about, this isn't going to gain every single overlap. There's going to be a lot of overlapping areas that you can't get because they're repetitive elements, and if they're repetitive, you'll never be able to use this methodology. I have a cat on me. I take no responsibility for what he does. I'm sorry. I'm very sorry, NMD. His booty is on me laptop. Excellent.
A process that took about 6 years. The cloned fragments were sequenced in labs around the world, following one of the project's two major principles. that collaboration on our shared heritage was open to all nations. In each case, the fragments were arbitrarily broken up into smaller overlapping pieces, about 1,000 base pairs long. Then, using a technology called the Sanger method, each piece was sequenced letter by letter. This rigorous map-based approach, called hierarchical shotgun sequencing, minimized the risk of misassembly, a huge hazard of sequencing genomes with many repetitive portions, like the human genome.
But there were still very difficulties on there. I'm here for a new reason. I'm trying to start a worm farm for my chickens and gardens greenhouse and fun. Very
In
cool.
dreams,
Blueberry
one is
sleep well,
not tethered by earthly limitations.
Very cool Christmas break.
What does that mean? Come.
Hey Pimp Cat, how you doing, Pimp Cat? Welcome in, Pimp Cat. Talking about this new study that came out that's sequencing many different human genomes. They're going to have a total of over 350 genomes that they can use for new comparative studies and better personalized medicine.
Consortium's better safe than sorry approach contrasted starkly with Celera's strategy, called whole genome shotgun sequencing. It hinged on skipping the mapping phase entirely, a faster though foolhardy approach according to some. The entire genome was directly chopped up into a giant heap of small overlapping bits. Once these bits were sequenced via the Sanger method, Celera would take the formidable risk of reconstructing the genome using just the overlaps. But
yeah, so Dame Karen, the other thing about the diversity too is that not only it's more increasing in the total number of people that they have, but what they'll do is that they will actually get other populations as well. So it's not just, let's say, college student population from Ivies, but rather all over the world, different populations. So you won't misidentify something as a disease state, but rather you actually have the comparison, like this is very unique to this and this is unique to that. So that's going to be a much better tool for it. Guys, go check out lovely and amazing Andrew CZ. Chat, Andrew CZ, breathtaking amazing soul. Love you, Andrew. I love you so much, Andrew CZ.
Perhaps their decision wasn't such a gamble, because guess whose freshly completed map was available online for free? The Human Genome Consortium, in accordance with the project's second major principle, which held that all of the project's data would be shared publicly within 24 hours of collection. So in 1998, scientists around the world were furiously sequencing lines of genetic code using the tried and true yet laborious Sanger method. Finally, after 3 exhausting years of continuous sequencing and assembling, the verdict was in. In February 2001, both groups simultaneously published working drafts of more than 90% of the human genome, several years ahead of the consortium's schedule. The race ended in a tie.
I would argue it didn't end in a tie because Celera wouldn't have been able to do it without the existing assembly, like without the actual map for it. Not ideal when the doctors give a rare immune mutation and realize you're just for it. Yes, Ariella, indeed. I mean, that's the example we were giving if someone was from California versus you're comparing it to someone in Japan or Australia, there's going to be differences in the genome that are not resulting in any kind of negative health effect. It's just base mutation. Without Celera, it would have taken human genome 7 years to do 3 years of work. But they did take them that long, right? So they didn't collaborate on it, Celera just took the existing data that the Human Genome Project already had and utilized it to interpret their own information. So they actually didn't utilize it for anything extra beyond that. By the way, now welcome in.
The Human Genome Project.
And Blueberry, there's no worries, but there's also no sorries, Blueberry. If there's sorries, that's what's going to happen. Japanese audio, your spine might be able to shatter in the next video. So it turns out you might just have Irish heritage. Yes, that's why we need them more on that front. I love that the guiding principle was sharing their map within 24 hours. Yep, but it didn't, and because they weren't doing it for profit. Without competition, would have stuck to the original timeline. No, I'm not saying that the competition isn't good. Competition's great, especially for innovation. It was just differences in how they approached it. I still think the Solarix one wasn't the better one in terms of the logical flow. Like, they wouldn't have been able to do it without the map. Yes, you got it, Blueberry. Have a good one, Blueberry. Go to sleep, Blueberry. Go to sleep.
Project's practice of immediately sharing its data was an unusual one. It is more typical for scientists to closely guard their data until they are able to analyze it and publish their conclusions. Instead, the Human Genome Project accelerated the pace of research and created an international collaboration on an unprecedented scale. Since then, robust investment in both the public and private sector has led to the identification of many disease-related genes and remarkable advances in sequencing technology. Today, a person's genome can be sequenced in just a few days. However, reading the genome is only the first step.
The few days has been lessened. Now it's just overnight. Overnight, you're actually able to sequence the genome, but the reading is still the difficulty.
We're a long way away from understanding what most of our genes do and how they are controlled. Those are some of the challenges for the next generation of ambitious research initiatives.
So that's the backstory of Dame Karen of how and what actually got to this level of infighting, collaboration, competition, all that like weird jazz that happened there. The next step, though, is actually how to interpret the human genome. So let's say we've got this whole like laundry list of A, T's, and C's, and G's. What does that actually mean? How do we interpret that? And that's where the new study will hopefully shed some light into it as well.
Do you know how interesting your genome is? Your genome is made of just 4 molecules that we code for with 4 letters, A, C, G, and T. There are about 3 billion letters in a human genome.
I'm actually not happy with the sound of this, y'all, so we're going to move to a different video. I will post that video onto the Discord, but again, the sound is just not coming out through loud enough, so we're just going to move on to the next batch. This is a little bit deeper look into the Pan Genome Project, which is, again, the paper we're talking about tonight.
On March 23, 1997, the Buffalo Evening News ran an ad for volunteers to participate in a very large international research effort. You may know it as the Human Genome Project. The goal of this 13-year and almost $3 billion scientific endeavor was to read out the blueprints of the human species. That is, the DNA making up the human genome. Researchers started with DNA samples from multiple anonymous volunteers. The largest amount came from someone who responded to the Buffalo Evening News ad. The end result was the generation of the first ever sequence of the human genome in 2003. Scientists call this the Human Genome Reference Sequence. By acting as a baseline to compare other genomes to, the draft human sequence have been pivotal for advancing our understanding of human evolution, how diseases are caused, and how the environment and heredity work together in defining our biology. But the human genome sequence...
Pimcat says, can you briefly explain how they find what's known as genetic markers, certain things like cancer, Alzheimer's, disease risk, especially since everyone's DNA is so different? How do, in other words, know what's normal? So Pimcat, that's why this new study is such a big deal. So currently what they do is they take potential genes of interest, Pimcat. So let's say for Fragile X, the gene FMR1, we know it has a mutation in it. So what they'll do to figure out if there's genetic markers, if someone has FMR1, they'll compare that particular genetic sequence to one another, to a patient who we know doesn't have it, to a patient that does have it. And then they're looking for differences in that one particular gene. And those are genetic markers that could be a risk factor for that disease. Same thing in Alzheimer's as well. You can take brain samples from Alzheimer's patients, compare them to patients without Alzheimer's, and see what the differences are in known genetic regions. That interest, like regions of genetic genes that you are interested in because there's some evidence to suggest that they are neuron specific, for example, for Alzheimer's. Now, PIMCAT, one difficulty is that researchers have been doing these comparisons of a whole genome against a single reference genome. So what we deem as normal has all been built against this one genome. And what's difficult about that is one person's normal might not be another person's normal. There might be mutations in certain genes that are not risk factors or not genetic markers for a risk factor. It is just variance. And that variance is almost meaningless. There's ones too that you might think are meaningless, but are not. Some genetic mutations actually cause differences in the 3D shape of DNA of your chromatin, and that can yield a disease risk. Right? So it's complexity upon complexity upon complexity. So what you want to do, PIMCAT, is you want to have multiple reference genomes. That's what this study is going to be looking at. So they're getting 350 human genomes fully sequenced as additional references. And from there, you can get the aggregate answer to what is normal and what is a mutant state. And then you can sequence a particular patient's genome and compare it against those references. And then you can figure out, okay, this patient has Alzheimer's, here, and here is where it's mutated, like in the genome. Let me compare it to 6 other Alzheimer's patients, and then you can see, okay, of the 6 patients, all 6 of them have a mutation at this one particular site. That might mean that it's a genetic marker for Alzheimer's. Does that make sense, PIMCAT? You're essentially taking a law of averages. You're not testing any of these in humans. Instead, what you end up doing is you might put it into a different organism, that particular mutation into a mouse, a rat, or a fruit fly, a nematode, a yeast cell, depending on what question you're asking, and then you see what the effect is, if that yields a mutation or not, like a physical change inside the animal. Does that make sense, PIMCAT? Hi, Crux. Welcome in. Happy vacation out for me. I've been hanging with the family. And in some company meetings today, I've enjoyed so far. Excellent, Crux. Are you at the beach, Crux? I can't remember. It is not Chuck Norris, unfortunately. Is this part of the testing for IVF to figure out what can be transferred to the baby? So with IVF, it's similar, Coffee Rocks. There are and you can do this actually for not just an IVF test, but an amniocentesis. So testing to see the baby's genetics up against a panel of known diseases and seeing like, okay, they don't sequence the whole genome of the baby, Coffee Rocks. They just sequence little sections here and there and compare it to a reference panel. And if in the reference panel there's a match, that might put that baby at risk of having X, Y, or Z. And so again, that's not whole genome, that's very, very narrow windows. Sounds like it's specific sequences that look out of the ordinary. Yes, PIMCAT. So interestingly enough, PIMCAT. Clap Slap! Thank you for the follow, Clap Slap. Welcome to the colony, Clap Slap. Interestingly enough, PIMCAT, there can be mutations that yield 3D changes in the DNA, and the 3D changes in structure can have an effect on how much a gene is turned on or off. That doesn't get really seen. Ariel, thank you for hydrate.
What drink?
That does not get seen in just this like particular sequence study. Instead, you have to do the whole genome sequencing for changes in the 3D architecture. And there are disease states that are influenced by that. There's a gene called MYC, M-Y-C, that's usually mutated in cancer. And it's an extremely long gene. And for a long time, folks thought it really had nothing to do with anything. It turns out it loops in on itself. And because of that looping, there's a gene inside of that whole region. And that touches other chromatin regions and turns them on and off. And that's why it's important in a lot of these disease states. And you never would catch that if it wasn't for this 3D sequencing that they can do. Ariel, I'm not sure when my next blog draw is. Sometime next week. But I appreciate you. 3 more days off on vacation until back to work. I hope you enjoy your vacation, Crux. I hope you enjoy it. If you're looking for a specific marker, do they isolate something or do the whole genome? If you know what you're looking for, NMD, let's say if it's a mutation in a known gene and you're trying to see if you have that mutation that might predispose you to Alzheimer's, they look at a particular region, NMD. I'll show you how to do it. Just one moment. Okie dokie, artichoke. So NMD, what you'll end up doing... Toasted sandwich! What's on the sandwich, Alex Vixen? What's on the sandwich, Alex Vixen? Good news, we know what it does. Bad news, we have a hulked out... I mean, that's not a... That would sound like a pretty cool movie, Alex. So NMD, let's say that this is your genome right here, and the gene that we're interested in is here in blue. You don't want to do the entire genome, NMD, because it's much more expensive. So instead... If you know that this is your gene of interest, that's the gene that we want to figure out if there's any mutation in there or not. You know the beginning of the gene, and you know the end of the gene. So what you can do is you can chop up the gene, the genome, and you can add adapters to your sequence of interest because you know what the sequences are at the tips of the gene. So I know that the start site here is of these certain base numbers. Hello, Monk. Welcome in, Monk. How you doing, Monk? And then this side too, NMD, I know what the sequence is. I just don't know the sequence in the middle, especially if the sequence in the middle has any changes. And so what happens, NMD, is this will sequence, this will copy, and then this will copy as well, and you'll then have 2 copies of the gene as DNA, and then you keep doing it over and over and over again until it just exponentially goes up. So the total amount of that DNA sequence that you have will be boop, like a huge amount. So genetic cause would see if it had the BRCA gene. This is what they did. This is the exact same thing they did, NMD. This, like, imagine in your genome, this is the BRCA gene. Not BRCA, like Mortal Kombat, but BRCA. Important in breast cancer. And so it's actually like 80% predictive if you have a certain mutation. So what they would do, NMD, is they would put the adapters at each of the tips and then sequence it. Like then it just replicates as it goes in, and you replicate it over and over and over and over again until you have a whole jar full of this particular gene, NMD. Essentially, you get rid of all the excess stuff, and then you put it into a sequencer and you figure out, okay, what are the actual reads on there? What are the actual base pairs? If NMD, let's say the normal one is AATCG, let's just say for the sake of argument, and yours comes back as AATCG, then okay, you're set. But let's say, NMD, that there's somewhere in this genome where we know that if you have a particular mutation at this site, that's the really bad mutation. And so then again, they'll sequence you and they'll look and see and do a little bit of comparison. And when they get the comparison, if you have that red letter there, that would be indicative that you do have the mutation. And that's how they do it for a lot of things, NMD. So, as Coffee Rocks asked too, I think they screen for over 500 different things. Like even an amniocentesis, you can screen for in a newborn. You can also have genetic counselors pre-screen you before you have a kid, whether it be for IVF or regular F. And they'll tell you what the likelihood is of getting this certain kind of disease state, and if you should be screening an embryo or not because of risks that you may have. If you have a gene marker, will you have it from birth or could it change over time? It changes usually. It's both, Bit Gaming. So especially it's difficult with cancer, Bit Gaming, because usually right when you're born, your genes that are mutant for cancer are usually fine at the time of birth. It's usually environmental factors that can induce mutations will yield that particular effect. So like in a tumor, those tumor cells are very different from your other body cells. So the particular side of it was happening. So you can get mutations just, being out in the sun and not wearing sunscreen. You could expose yourself to UV light. You can do a bunch of different things. Or you can also have the mutation at birth as well. And, having the mutation, the BRCA gene, doesn't mean that you're going to have breast cancer. It's just I think it's NMD, correct me if I'm wrong, I think it's like an 80% likelihood that you will have it. But it's not definitive. So some things are at birth bit gaming, and some things, and yes, like effectively you'd be fine at birth, but it just doesn't manifest until you're older, like as you age, that it actually yields an effect. So it could be many different factors. You didn't have an excellent NMD. I believe it's a very high winner. That really famous astrophysicist, STEM scientist, Serafina Nance, had it, and so she had a double mammectomy to prevent her getting breast cancer, and she was still in graduate school when she did that. Just because she's like, I just don't want cancer. It's kind of wild. Coffee Rock says like Down syndrome or nubile tubule defects. So Coffee, for things like trisomy 21, the 3 different chromosomes, you could see that without the big genetic sequencing. It shows up in the sequencing as well, but that's like a standard karyotype, because you'd see whole differences in chromosome numbers. You'd have 3 of 21 instead of 2. And then for particular syndromes, you'd see it in one of these sequencing maps. You can test yourself at birth to see baseline, then as an adult and see how it changes. You could. It's hard, Coffee Rocks, was as you age, because it also could be certain populations of cells that are changing. So like if you are smoking a lot, smoking is carcinogenic, which means it causes mutations in your lung cells. So Coffee, if you smoked a lot and then took a skin cell sample, and when you're older, those cells might not have the cancer predeterminate mutation, but maybe they have it. Maybe they have it in the lungs, because that's where you're exposing yourself to those mutations. Does that make sense, Coffee? So it's harder as an adult, again, just because of where your changes are going. I would imagine, Dame Karen, they would do amniocentesis for Downs. I would imagine they still do it for any kind of genetic change. I believe 60 to 80%. Awesome. Thank you, Smikes. Not awesome that it's something, but thank you, Smikes, you handsome devil. Smikes knows things. Smikes, 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 Smikes. Awesome. Excellent. Glad to hear it, Coffee. Okay. Smikes. Thank you, NMD. I'm sorry. It's too fun to push that button. It's a test that they will offer you, but you can tell them to. You can. I think also, Alex, they can ask for Smikes. You can ask for subsets of the data as well. I think they give you, like I said, 500 plus things that they screen for. It also depends on if you see a genetic counselor beforehand. That they're not looking for things like Downs. They're looking for combinations of genes that could yield states of like certain amounts of sickness, like blood disorders, heart disorders, things like that. Of course, they absolutely can, Dame Karen. Many people can live long and good lives. Yeah. Christmas Bricker, indeed. What on earth is that? Push the button now. I see. I had no idea what that emote was. Okay, let's continue the Human Pangenome Project.
Sequence generated wasn't quite complete. While the sequence reflected about 95% of the human genome, 5% was unknown, regions called gaps. Since 2003, researchers have been methodically filling in those gaps. And today, there are only around 100 left, down from 150,000.
So the gaps were over 2 decades almost in the making, and they still... Actually, they are over 2 decades trying to fix up what those gaps are to have the actual whole human genome sequenced.
But why are there still gaps in the human genome reference sequence? One major reason is technology. The machines that read DNA cannot read all 3 billion letters in the human genome at once. When they try to read too much DNA in one go, they tend to make more mistakes. Like trying to understand one really, really long sentence at once. The remaining 100 gaps mostly contain long stretches of DNA sequences repeated over and over again.
But like we talked about earlier, the repeating elements, there is new kinds of long-form sequence reading that they're trying to do to fill up those sequence reads, and hopefully that'll eliminate the gaps in these new samples as well. I've been listening. Oh, gotcha. I got you, Chris McBricker. I got you. Appreciate you, my friend.
These regions have turned out to be exceptionally difficult to sequence. Back in the 90s and 2000s, older DNA sequencing technologies couldn't determine if such sequences were repeated 10, 20, or 50 times. But researchers did not give up. They knew there was a possibility that such repeated DNA regions could contain genomic information relevant to human health and disease. But generating a truly complete human genome sequence is only part of the challenge. Even after the human genome reference sequence is made gapless, you can't represent all of humanity with just one reference. But why? Why isn't one human genome sequence enough? While the genome sequence among all humans is 99.9% identical, that other 0.1% accounts for all of the genomic differences in the human species. And populations from different ancestries have genomic variations that may be specific to each group, which is why one human genome reference sequence won't reflect all of these different genomic variations. Researchers need a range of reference sequences that encompass the genomic differences in any ancestrally diverse population. Put together, they would showcase all human genomic diversity. Unfortunately, up till now, most data about genomic variants that may be involved in different diseases have been gathered from people of European ancestry. But that's
not the only thing. It's a lot of it is college students because they're easy populations to get a hold of. You can offer them a couple of bucks and they'll do it. Other 1.1% is moon people. Big gamey moon people. Oh my God, Luna. Oh, Dame Karen. Very nicely done, Dame Karen. Very nicely done, Dame Karen.
It's just one ancestral group. This means we may be making some medical decisions for other ancestral groups based on incompatible data. But scientific efforts are underway to eliminate this historical bias. One such effort is the Human Genome Reference Program, funded by the National Human Genome Research Institute. Over the next couple of years, at least 350 high-quality, gapless human genome sequences will be generated from ancestrally diverse people. Remember those gaps in the genome? Another group of international scientists called the Telomere to Telomere Consortium are...
The baseline would change. Yeah, they have to keep doing these sequences, Smikes. One nice part about it is the price does keep going down, and eventually the technology is going to keep getting better and better, and so all the gaps and the speed and the price will continue to go down, so you can keep adding to that consortium. I think that would be the best thing to do. You $20 and a Big Mac if you let us take a pint of your blood. Big Mac, meal, Alex, you're not wrong. Face matching that last one made me think one side was an ape and the other a human. They're looking at the evolutionary trends of it, Zen, which is also nice because there's actually many more ape samples that are sequenced that are human ones. So that would be a cool thing too, Zen, is to use this tool from an evolutionary perspective. I think that would be really neat.
Working to fill them in. And
Nerduino, he lives, chat. Nerduino lives, folks. If you're not, NASA is in his house, as Gwendolyn P says. Guys, go check out the amazing Nerduino. How you doing, nerd? Nerduino, we missed you. Doing all right, Nerduino. Doing all right, my friend. Doing all right. We're talking about this new study that came out looking at 350 human genome sequences and doing comparative analysis between them. How many sequences do you need to have statistically relevant sample size? So it's depends on the question that you're trying to ask, Smikes, which is the unfortunate out that I'm giving myself. if you're looking for differences in populations, you're going to need more than what they have. And you'd have to have many more populations. You'd have to have in between populations, and you'd have to have a much more depth in there as well. And same, if you're looking for disease states, then it's also a different question of populations, right? Because you'd want to be having something that's either wild type for that disease state you're asking about, so then you can actually look and compare against what you know has a disease state. So how many? I don't know, Smikes. There's these things called power analyses that you can do. So it's a predictive way of figuring out, in order for your statistical test to have sufficient statistical power, how many samples do you require? To me, Smikes, that's a load of baloney, that kind of statistical test, the power analysis. Although there's many people swear by it. I did some of these analyses in grad school to like ask how many experiments to do to have something be significant, and it never made any sense. But yeah, I don't know, Spike. I don't know. Demon Speed, thank you for lurking. A good question, Spike. That's probably even a Lita question, it would depend a lot on the question that you're trying to ask with the data. Here's a question: could be 350 people and 350 sequences. Could you tell me with which? Maybe, Nerduino. If there's enough differences between them, we might be able to. Somewhere between a lot and all of them. You're not wrong, Alex. It's just dependent on the question that you're trying to ask, whether you need more or less power.
Generate totally complete sequences with improved technologies. Using these 350 complete genomes, the Human Genome Reference Program will then combine the data and create a unified genomic representation of the human species, calling it a pan-genome. Will this be enough to serve as a single reference for all human genomics research in the future? Perhaps not. But so long as these people are thoughtfully chosen, this effort will go a long way towards that goal. Slowly but surely, we are moving closer to one of the fundamental aims of the Human Genome Project: a complete and accurate representation of the genomic blueprint of the human species.
Do we know who this first sequence belonged to? So, Smikes, here's the funny part. It wasn't just one person. It was 70% of one individual, and then other pieces from other individuals as well. So that's actually a fun, not so often talked about thing that reference genome is actually an amalgamation of a couple of different individuals. Yeah, it's not just one person, which also then makes it a lot harder, Smikes, because you're not just comparing to a lone individual that you can then look at and see what their phenotype is, right? Like, they don't have these things, but we can tell based on their genetics. It's a mix of a couple of individuals. It's not ideal, but it's the best that they could do at the time. Modern day, you never have that, but at the time, it kind of made sense to have something like that. Okay, so that, folks, was paper number 1. So they've actually released 47 people's genomes that they've gone ahead and sequenced, and they're increasing that to 350 by mid-2024. So we will check in, hopefully mid-2024, on what the status is for the additional sequences of how many genomes have been identified, because that'll really yield a nice extra bit of information to many people. And then again here, this is the video we actually just watched. So this is from the group themselves, explaining of what they're doing and why they're doing it. And it's got a really nice little milestones chart as well. Rainbow and CC. What is Rainbow and CC, Alex Fixon? Bentley, I see you there. Bentley, you're not getting away without one of these. Guys, go check out Love Amazing Bentley. Hi, Bentley. So on the website, they actually go through and look and see what their year 1 goals are, the current productions, and what the final completed goal is as well. Completely agree to understand the genome and fix what we can, but diversity also helps new diseases figure out what it has in store for us. Chris McBricker, absolutely. They're not saying change the whole genome. They're saying just change single mutations that could be disease causing. You don't want to nerf all the entire genome of diversity, because like you said, if there's different diseases that come about, there goes our protection against them, because we need genetic protection for it. Yeah. So I think it's a nice little map that they have what they've done so far. Rainbow was the first cat to be cloned. CC, copycat or carbon copy. Oh, that's quite clever, Alex. I like that. That's a very clever name for it. And then here are all the collaborators as well. So there's universities across the United States and Europe as well that are participating in this project. So Dame Karen almost has feels of the original Human Genome Project in terms of all the collaborations that are occurring on this. So you have East Coast, Midwest. We have if Ariella is still here, Ariella's institution right there, Washington University in St. Louis. West Coast ones, there's also some companies as well. And then you have like the Sanger Sequencing Company here. They're also the Embl. Yeah, all the different ones that are working together. Guys, please go also follow Ariella Studies. Please go follow lovely Ariella. Gotta give me 5 more seconds, Ariella. Dana-Farber. Yes. Yes, that is also there. Hi, Daybreak Stations. AKA Daybreak Studios. How you doing, Daybreak? Who else wants Daybreak? So that's paper number 1. Now, I promised y'all paper number 2 was going to be very close to that. Paper number 2 tonight is published in the journal Nature, which is one of Smike's favorite journals. It is called A Comparative Genomics Multi-Tool for Scientific Discovery and Conservation. This is a really awesome another genomics study, but it's not focused on humans. It's instead focused on animals. And it is, if you look and see...
This is a massive amount of scientific data.
If you look and see who it's by the Zoonomia Consortium. So the Zoonomia Consortium is a giant list of people.
Gucci.
They're pretty Gucci, Nerduino. Let's see. Here are all the people on this paper. Is this why animals that usually reproduce asexually also have the capability to produce sexually reproduce for diversity? Yes. So Coffee Rocks, when we talk about the nematodes, they're usually hermaphroditic. So they are self-fertilizing. In times of stress, they will produce males to mate with, and that introduces genetic diversity. Absolutely, that's the goal, yeah. With Zootopia, listen, Big Gamey. Did you hear, Big Gamey, about that there's gonna be a Zootopia 2 Electric Boogaloo? Did you know this, Big Gamey? There's a Zootopia 2. I really like the first Zootopia. I think the sloth at the DMV is hilarious and it's very on point. It is very, very on point, Big Gamey. Flash, yes, yes. What do they eat? The sloths? I'm not sure. Heard a whisper. I'm looking forward to live action Moana with The Rock. Why? Bit gamey. They just had it. I love the Electric Boogaloo in my childhood. Oh. What do they eat in Zootopia? I have no idea what they eat in Zootopia. That is a great question, Smike, because you can imagine perhaps a cannibalistic world where they're eating left, right, and center. Why should not be the question? Well, the answer to the why, Big Gamey, is coin. Hi, Black Sheep Meat. Welcome in. Smikes, they cannot answer anything about this in this paper about diet. What they are looking in this paper, by the way, NMD 96 is in this paper. Let's just quickly read. Here, I'll tell you all about the paper. What they did was they wanted to do a huge survey of mammals and look at what their genetics are like. And so what they did, they took 240 placental animals, so mammals that have a placenta when they're, carrying their offspring. They took 240 of these mammals. It's only 4% of all mammals on this planet, but it's still 4% more than what they had before. And they went in and they performed genome sequencing on these animals. And so from 240 animals, they got genetic sequences. So here are some of the animals that they tested. And you can see at the top, right here is NMD96, right here. In Zootopia, their diet is primarily fruits and veggies. I think the carnines get synthetic meat. I cannot remember. So went down a rabbit hole researching how ants drink water because they decided to take rehydration but direct more creative attention. Ariella, they have these little like Slurpee tubules almost. They go. Yep, and they help with these little tongues.
Where's my drink?
Through capillary action.
What
Okay,
drink?
Cool. Science says my brain is like a sponge, absorbs all knowledge, and like G.I. Joe says, knowledge is half the battle. But Hugin, if knowledge is power and power corrupts absolutely, then doesn't knowledge potentially corrupt you? Hugin, let me know if you have the answer on this. Big Amy, I'm about to do something terrible. It's empty, Big Gamey. It's empty! Only a Sith deals in absolutes. Hugin, is that not by definition an absolute? By you telling me the Sith deal in absolutes and only them deal in absolutes, that's in fact an absolute, making you in fact a Sith, Hugin. I'm gonna go get some water.
Bullet the science guy.
The science guy. Bullet.
Balin the science guy.
Science rules.
Balin the science guy.
SciAnts is a partner on Twitch. With lovely wife Lita.
Balin the science guy.
Thank you, Big Amy. And Alex, my manager as well. I do dress in all black and cast my fear upon my enemies, so would I be a Sith or am I just Batman? No, I think Batman deals in the grays. I think you're fine. I think you're alright, Hugin. I think you're okay, Hugin. So, chat. This is another genomics study. So they are looking at a bunch of different animals, and they're comparing and contrasting their genetics and trying to figure out evolutionary patterns that were not visible before. So they have in red here is from this Zoonomia study. In gray is in this GenBank, which is whenever you sequence an animal's genome or do any kind of genetic sequencing, you always deposit your sequence into GenBank. That therefore allows you to essentially back up your data. In this peach color is where it's both in Zoonomia and GenBank, and then in white are some missing animals that they're looking for at. They're going to sequence these animals' genomes in the next series of studies. Welcome in, Celadon K. How you doing, Celadon K? Welcome in, Celadon. And yes, Smikes is on point with this, chat. Smikes is so on point, chat, that I wish I had an animation for a golden sticker or something, but I just got why am I pointing out what Smike's saying is such a big deal? Because we just talked about the reason that having just one human sample is not great is because there's nothing that we can really do with that. There's not really anything that you can compare against. You can't figure out what's normal and what's abnormal. However, that is not the question they're necessarily going for here. What they're going for in this study, Smikes, is more just a broad range of evolutionary topics. Like maybe how does evolution work across a broad series of organisms? And so you're not going to be looking at anything independent and like all by itself of like how these different things work, but rather as like a global image of like, okay, if this evolved at this rate and this is evolving at this other rate, then maybe what genes are undergoing a lot of extra changes that we wouldn't have known about. And so it's things like that that are now going to be able to be answered by this. So broad evolutionary questions can be answered by this, but beyond that, you can't get really into the nitty-gritty of it. You can't get too much into like all of those little extra bells and whistles, as it were, that you need many more what's the best way of putting it? You need many more sequences to be able to do any internal or differential components there. How does someone sequence a genome? What does it involve? Caledon, that is a great question. Caledon, you got to check the VOD for that one. We went through, watched a couple of videos talking about, and we were dissecting how to do genome-wide sequencing. The whole first part of the stream. It will be up on the YouTube, Caledon. Normally, I would answer you, but it is a very difficult question. It is not a straightforward thing, and it just takes a lot of like extra dissection of it. So that's not me trying to avoid the question. I just think it's maybe better for the VOD on that front. Are plant ones easier to genome sequence? So Smikes, plants are weird. Plants have things like overly copied, like extra copies of their genomes all over the place and beyond, and it does not make any sense as to how many, like they're much more plastic in terms of like how many genes that they have and how many copies of their genome they can tolerate, and because of that, having the different number of genomes and all these different copies can really influence it's comparative genomics is just much harder because of that. Tea Time Cat, thank you for thanking Risto for the gift sub. Hi, Mom Boss Lady. Welcome in, Mom Boss Lady. So let's actually talk a little bit about what information they got from this study, and we'll also watch some videos on it. They got 5 really, really cool study pieces of information. So based on this, let's see, one of them, they also had some dogs that they sequenced. And so they also got, do y'all remember Balto, the sled dog that was delivering medicine to children in Alaska? So Balto, it turns out, they sequenced some of his DNA and compared it to this reference genome. And what they actually found is that based on the comparison to the reference genome, it looked like Balto had an enhanced ability to digest starch. And also had, like, it was pretty clean in terms of mutation. And so that meant that he was just better able to survive in the cold. Which is wild they were able to do that. Yes, the hero Balto, exactly. Monk, yes, these are animal genomes now that we're talking about. They also found that he was very much not inbred. So Balto, named Karen, you'd be very hard to come by. There is no Balto breed, which, based on the cartoon, I never would have thought there was no Balto breed, because it very much looked like there may have been a breed of Balto. But no, apparently it's very genetically diverse, and that was in part another reason why he was able to survive the trip. Another something they identified was because they have so many different animals here, they have a lot of evolutionary time here as well. What they discovered was it seems like the origin of placental mammals traces back to 102 million years ago based on all this new data. So before this data, they thought that placental mammals were roughly 60 million years ago when they started to arise. This backs it up to 120 million years ago. And so this suggests that mammals had much more time to arise than just after the loss of the dinosaurs from the asteroid strike. This suggests that there were mammals there much, much, much earlier. Our Discord, yeah, Ariella, whatever you want, Ariella. You're Gucci. You're Gucci, Ariella. So that was number 2, which was very cool. Marsupials were more common during... They were Smikes, but it seems like the existing placental mammals were also more common than we thought, which is kind of wild to me. Another thing that they identified in this study was that all these genomes could be helped to identify cancer-related genes. So one thing they identified, they're talking about, is maybe they can find which genes are not as resilient to mutation. Which genes, when you have mutations and changes in, could yield negative effects or just not likely to pop out. Which genes are undergoing evolution at a much higher rate. So it's this weird measurement called DNDS. There's the ratio of non-synonymous mutations to synonymous mutations. Synonymous means if we mutate these, there's no differences in the actual DNA. And then if there is mutation, that there is a difference in DNA, that's called non-synonymous. Regions undergoing rapid evolution are not under can be under selective pressure for it, and so those usually aren't regions that are undergoing like cancer related things. Gerhard Sparks, I need to find a hobby. Welcome in, Gerhard. How are you doing today? Welcome on in. They also identified more jumping genes in this study. So those are genes that hop around in the genome and move around, and they usually make up 75% of the animal's genome. Interestingly enough, there are, you can see on this map, carnivores and herbivores. Indeed, Daybreak. Carnivores and herbivores, and they find with carnivores, there's more jumping genes active than there are in herbivores. And this seemed like it's very, very well represented in this tree of life. So that suggests that based on what you eat, your diet affects your genome and its evolution, which is kind of wild to me. The 5th big thing they took out of this was that if you using the comparison between humans and other mammals, you can do some neuroarchitecture and brain studies as well. So they did a lot of comparisons between like neuronal regions and saw what's evolving faster in humans versus other animals. And because the way the human brain works and the size of the human brain, they're wanting to compare that brain plus the brains of things like dolphins and elephants and other of these animals to figure out maybe what genes are most important for human brain evolution. So it's a huge smattering of genetic information that we're having our hands at. All right, Ariella, what is this? Oh, open in the wrong window. Ariella, that link does not like me. That link does not like me, ma'am. Hold on. Ooh. That link just opened me up in Discord, and I don't think that's what you wanted. Aha. I got it. Beautiful, Ariella. It was no, Smikes, it opened up, it tried to open Discord for me instead. Gerhard, thank you for the follow. Welcome on in. I like looking at him not making art. Gerhard, making art is lovely, though, I'd have to say. This is the one from our friend Ariella Studies who's in here, making a beautiful piece of anti-hydration. The caption is, don't forget your daily moisture wicking. Y'all, that's adorable. I can imagine moon cows now, super huge and great for cheese. Chris McBricker. That sounds delicious. That sounds absolutely delicious. And Ariel, this is beautiful. Thank you so much, ma'am. I also like the signature. Is this an uwu, Ariella? Ariella, tell me that you don't do the finger thing, though. Do you also do the finger thing, Ariella? This thing? It is an uwu, but is it this one? Is it this kind of uwu, or is it a regular uwu? Thank God, Ariella. Okay, good. I'm fine with this. Ariella, a lot of people, when they do the uwu, they go like this. And they go uwu. It's Ariella, if you look online and you see those emojis with the fingers touching one another, it's a whole uwu thing. It's a whole uwu thing. I don't even know why, Ariella. No, sorry, Coffee Rocks. The ants are going. They're still kicking coffee. No, Tarzan, how dare. There's no news yet. There's no news yet, coffee, from the vendors about a new ants. Like, you get a new colony. Unfortunately, too, coffee, it didn't help because I had to bring them all upstairs because of the flooded basement, and so they're not happy in their, like, little temporary container, so... And I don't yet have the final quotes from everyone, so it hasn't even gotten started to be fixed yet. It's a whole thing. Hoping, though, that I'll be able to get it. We'll be able to get it fixed soon. Okay, so again, here's the paper, and let's actually watch a few videos on this Zoonomia, because I think it's really cool. And again, this, I hope y'all are kind of enjoying tonight. I know Genomics can be a little dry, but I hope y'all are appreciating, like, all the things that we can do with all these genomes. Not just all the extra human genomes, but also all the extra animal genomes. Being able to do a bunch of, like, evolution studies on these things. And just, like, disease studies. It has a huge potential. Like, there's a boatload of information in these studies that I hope y'all are appreciating, just like how cool it is. Nature must have charged a fortune to publish that graphic. Nature no longer does that, Smikes. There are still journals. This is interesting, y'all. There are journals that charge you for color and black and white figures, even though it's black and white. Or, I'm sorry, even though it's just in print. Or not in print. It's only online. There is no print version. Right? This made sense when they were printing the copies of the publications, but now that everything's just online, it does not make sense as to why there's these different things. And so Nature just instead charges you 10 grand, Smikes, to publish it. There are journals that still charge based on color figures or black and white figures, and black and white's cheaper. And it's like, are you charging your like some kind of like fee for the pixels? It's just a scam is what it is. Might be a Disney person, Mom Boss Lady.
Data. It's literally terabytes of data that's actually behind this in a database for scientists to go and download. How could we package that, and how could we actually make that look really interesting and engaging? The Zoonomia project is for scientists who are sequencing genomes across the mammal spectrum, and what they're looking for across the genome are traits and characteristics that are common and different across mammal species over time.
And so the primary goal for this was to provide access to the data empowered of comparative genomics as a tool for human medicine development and biodiversity conservation.
So we thought, how could we actually start to combine this very sort of contemporary looking modern take on genome sequencing, which is the most cutting edge scientific technology, and very old looking prints.
Is there anything on rabbits? So Monk, there are rabbits in the study. There's even an image of one of them. So there's the snowshoe hare here. That is one of the animals that was genetically sequenced. So Monk, it is Science News of the Week, and we're talking about a lot of genomics tonight. So a lot of genome sequencing and comparing these genomes into between different animals and different humans as well. No worries at all. I'm so sorry that you're nauseated, Pixels. Have a good rest of your evening. Please feel free better soon. Feel better soon, my goodness. Shark and Raft is so kind. It tells you not to use that particular corner. Yo, Zen, that's adorable, Zen. I think. Guys, let's go back to this video on the Zoonomia project. Not Zootopia, Zoonomia.
And bring those two parts together.
And then at the centerpiece of the site, we have this mammalian tree. So it allows users to kind of glance at the scope of all mammals.
So this is a giant tree of life here. And each of these little lines is a different animal. So that's the 240 plus animals that they genome sequenced. And if you zoom in on it, you can actually figure out like the branching pattern and where they're related and how they're related, and then do comparative genomics between these different animals. So you can compare an anteater to an ape, to a human, to a bat, and so much more. It's really, really cool.
So we're kind of letting the scientific information and what the scientists wanted to say with the website drive the design direction, and then looking at how we could integrate technology into that.
I think the biggest breakthrough was finding the ITOL annotation.
Daybreak, you could, not in this tool. This tool is only mammals. But you can. There are ants with genomes sequenced. You couldn't use their tool, but what you could do is you could download the sequence of the anteater, and then you could download the sequence of the ant from a different database, and then do the comparison between the two. You would just need to have the training to do it. So the nice thing about this Daybreak is it has an interface that you don't have to be a programmer to understand. But if you're starting to do these kind of comparisons that are outside the scope of what they have, then you will have to be able to do that. My aunt had her genome sequence. She bags it out at Thanksgiving. Nice, Forever Grasshopper. This is just the tree of life we use, but with genomal comparison. Yeah, so it's the tree of life that we use, Smikes, but you can do a whole bunch of comparisons that you couldn't have done elsewhere, like in just the regular old tree of life. You could use a different tool. Smikes, a lot of these tools take the data of certain kind of formatting that they don't really tell you. It's kind of like the proprietary ins and outs of it. So everything is slightly different enough where it's not just an easy pop in and out, like import export kind of thing. There are base tools. If you can get the FASTA file, which is the raw sequence file, then you can run that through program in R or Python that analyzes the genomes. But it's a lot of these databases have already post-processed data. Yeah, so it's not just it's not just that. It's not just like a CSV or something at that point, Smikes. It's a lot of post-processing goes in there as well, unfortunately. Birian! Hello, Birian. How are you doing today? Happy Friday to you as well. Or happy Saturday now. At least for me anyway, it's Saturday.
Editor. Through that, we were able to adjust and style the tree by simply just updating a spreadsheet. And this was great because we're sort of designing the site at the same time as they're finishing up their research. So we're adding a mammal here, changing the assembly source of a mammal there. So all of this was changing rapidly, and that was just a huge discovery because it would have taken a whole lot more time to draw something that scale in Illustrator. The WordPress is a great tool. It's something that we use for nearly all of our sites. You know, we always develop the tech stack based on the desired outcome of the project. And in this case, it was to create something that was user-friendly and eye-catching.
Sadly, a lot of mammals are endangered around the world. So the impact that a project that this has is giving scientists access to mammals, to animals, and the genomes of those animals that sadly may not be around in this millennium.
I'm curious to see.
One thing I will say, Smikes, is that the user high tactics. The usability in terms of like an interface for non-experts is usually quite poor in between the developers do a really good job making this program, but then the interface is terrible and it's really difficult to use and it makes no intuitive sense. And then like this front end stuff is also then very difficult. So it seems like there's a couple of disconnect steps in here. I got to do some of the UI work for some of the databases we made and some of the testing we did, because I was like, I went to the lead guy. I was like, Casey, this doesn't make any sense at all. And he's like, what do you mean? This makes total sense. I'm like, no. I'm like, based on this, I have no idea how to use your software. And he's like, oh, we really need to fix this. Yes, we do. Please need to fix this. Tactics. It's been very genomics heavy today. So we've been talking a lot about genome sequencing, comparing genomes between different animals and then different humans as well. And so it's now like a little bit of the back end analysis of how all that works. So a little genomics feels a little devy to me, but it's I don't know. I hope it's been quite interesting for people.
If Zoonomia and the data that's available there might help influence some other research projects and maybe figuring out some of the mysteries of 2020.
Okay, so that's just a little intro.
In the International Zoonomia project, researchers have sequenced the genomes of 240 different mammal species.
And this is the new update for it. It's how they code for us. Exactly, Dame Karen. Tried to do gene ontology analysis a few times and it makes me cry. Logically incomprehensible UI. Ariella, the best one to do comparisons, unfortunately, also doesn't have a really useful interface. It's usually R-based programming to run through. It's the cuff diff suite, the cufflinks, and it's just you wrote your code. Okay, so you know the R code then. Yeah, unfortunately, the interfaces are quite bad a lot of times. The DNA arc is awesome. We were meant to explore and spread. I'm in ads. Gotcha, Chris McBrecker.
Professor Kerstin Lindblad too started the project in 2015.
And so this is another cool reason I was going to highlight the project, is because this project started in 2015 and only now in 2023 did they get all the genome sequenced. And the paper came out about 2 weeks ago for the Zoonomia project.
In the Zoonomia project, we are trying to understand why mammals are so different. There are more than 6,000 species and they vary from the 2-gram bumblebee bat to large whales which weigh many tons. By looking at the Zoonomia data, looking at the genomes of these species, we can start to understand why they are so different and why they have adapted to different environments.
Matthew Christmas is another researcher in the group at Uppsala University.
So this was part of a large international project where we sequenced a wide range of mammal genomes, and then we produced what you call an alignment of those genomes. So that's where you sort of compare the genomes to identify regions that are shared among them and also regions that are different. And we did that for 240 different species, and those species represented over 80% of the mammalian families. So we managed to capture a huge amount of the diversity in mammals, and then we could use that alignment to answer these questions that we wanted to answer about what's shared in these amongst all mammals, but also what are the different parts that have led to the diversity that we see amongst mammals.
In the Zoonomia dataset, genomes that are the same, or positions in the genome that are the same, that has stayed the same for 100 million years, are likely to be functional.
So you can look at not only organismal evolution as a whole, but you can actually zone in on particular regions as well. Like how certain genes are evolving. David, my God, I hated Ariella. I hated doing David analysis. That is absolutely terrible. That interface, Ariella, I don't know how you feel about it, but it made me want to throw the computer out the window. We can absolutely do a dive one day on these different tools that can teach you about things. But yeah, Lita did a bunch of David analysis as well. And I just, again, it's a great tool for the end product, and it's again looking at like genetic prediction and conservation, but like how you get there is a very, very painful way of actually getting there. It's it breaks you, Ariel. It breaks you.
And only about 1% of the genome is protein coding, so making the proteins that we need to build up our body.
Yeah, Smikes, I actually met the guy who did that particular database, and I was like, hey, could you make this, like, we were friends enough where I could be like, could you make this better? And he was like, what do you mean? I'm like, it's not usable. And he's like, no, this is great. I'm like, can you show me how to use this? And he had trouble showing me how to use it. Here I was, no, Zen, you're extremely handsome, and you could never be a tool, Zen. You're just, Zen, you are what we call a legend, Zentouse. Yes, Coffee Rocks. We do have a Moco Loco t-shirt on, Coffee Rocks. And it has very minimal spit up on it today.
Gucci.
Very minimal spit up today.
But there's maybe 10 times as much elements that tell when and why and how proteins will be made. So with the Zoonomia data, we could really see what these elements are, and that will help us understand genome evolution. We were able to address a question about whether mammals diverged before the extinction of the dinosaurs. And our data shows very clearly that mammals started evolving before, while the dinosaurs were still around, and that they then continued to diverge when they got more space to evolve after the dinosaurs were gone.
The researchers studied adaptation to different environments.
So we could use our data set to really sort of look into particular characteristics of mammals and how they've evolved. So for example, how sense of smell varies across different mammal species. Brain size is another trait that we looked into, because you see a wide diversity in brain sizes among mammals. And also hibernation, which is quite a specific mammal characteristic. And by comparing mammals that hibernate to those that don't hibernate, we could really pinpoint the genes that are involved in the hibernation process and the adaptations that they have in their genomes that enable them to be hibernators.
And actually, we probably won't get to it tonight, but there is already a study that came from this, looking at how what is the genetic basis of hibernation. For example, you know, when we stop moving for a long time, we're more likely to get blood clots. So why can something like a bear hibernate and not have a ton of blood clots form? It turns out they actually used this data set and did comparisons between humans and bears, and actually found some key differences in certain proteins for clotting factors that actually influence the answer of why certain animals can hibernate and not die, which is quite remarkable, right? How the liquids are always so very pretty colors. People visit the lab, they're always like, why are those liquids clear more? Yes, Ariella. It also is frustrating because when you're doing experiments, you're just mixing together clear liquids.
We Gucci.
You have no idea if anything's working or not. Or there's anything even in there, right? So watching that paleontologist streamer, and we were talking about this in the context of dinosaurs. Was it paleontologizing, Ariella? Dr. Christmas, I get irrational joy from this. He is a very jolly gentleman. I thought most bears don't hibernate sort of an in-between state. It depends on... So Smikes, it depends on the state, the type of hibernation they're undergoing, and the season for it. Is my understanding of, based on this paper, but that could, I don't know. It sounds like it's murky, Smikes. Sounds like it's murky. Yes, they do get bones. Yes, Professor Anduza, is one of our besties. One of our besties. So I was so skeptical because definitely not seen anything humans or mice or any of the bones I've personally seen. That's true. You can't just cut our bone, Ariel, and see how old we are. But dinos, maybe.
The sense of smell, we could show that based on the number of genes that are involved in smell reception, we could show that, for example, elephants have one of the strongest senses of smell amongst mammals, whereas something like toothed whales, like killer whales or sperm whales, they live in an ocean environment. They don't need a sense of smell, and we can see that in their genes.
The Zoonomia project also tells us something about biodiversity and how to save endangered species.
So genomics is a very important tool for biodiversity and conservation.
Torpor, not hibernation. Alex, let me quickly just pull up the paper. and just double check what terminology they used. So as per this paper in Science, Alex, they say that bears, brown bears hibernate. Per this particular science paper. That's just the one. That's the one study I'm looking for, Alex. Looking at, Alex, and that's the one that they indicate that is hibernation there, and not so much. But you know, that's just the current on the brown bear study. That's what they're suggesting.
Efforts. What we could see was that the amount of variation in the genome is predictive of whether a species is likely to go extinct or not. And you can look at a lot of different parameters of the variation that you see and then help figure out which populations, for example, are the most likely to survive. So we've generated a humongous amount of data, and it's all available for everybody in the world, and they can look both at different types of mammalian evolution and also to use the information to understand human disease.
And then they have one more cool video talking about how this will help with diseases, like looking at animal genomes and identifying, again, why this is helpful, not just from an evolutionary perspective, but also from a disease perspective.
So we're trying to understand why humans inherit disease. Humans are the most studied mammal in the planet, but we still don't have...
Ariel, now I can't not see the different colored tubes. Hibernation was in colder times, Topher was in warmer climates, Florida bears don't hibernate, Alaska bears do. I can't remember now. I will double check before we talk about that study. I didn't realize there was the nomenclature would have had differences. No sorries, Ariella. Ariella, you're just gonna get nerfed again, ma'am, and it's gonna be I can't control what people do. The rule here of no sorries.
Have enough information to understand which parts of the genome are causing these problems. And so we're really looking for the regulatory elements that tell the cells how they interact together. Those are the bits we're targeting. So if we sequence 240 mammals...
Where's Cliff when we have a capybara? Finally, for Cliff Alster McLean, we have a capybara on the screen. The poor gentleman is not here to see.
240 mammals, we can look at the positions where those mammals are the same and predict that those are the regulatory elements. We then focus there.
Jennifer Meadows is one of the researchers in the Zoonomia project. The researchers have studied the genomes of 240 mammal species, including the human species. One of the goals is to study hereditary disease in humans and in animals.
It's been a massive collaboration across many, many continents. People have been donating samples from endangered species as well as less endangered species like cats and dogs. And then we align the genomes, which is a massive computational task. So we're combining this constraint, or these regions that are conserved across mammals, with all of the data that's available for humans. So we can understand how frequently they change across populations. We know some...
Sleep well, Tarzan. Thank you for hanging with us.
...positions that make you sick.
Hi, Valiant Cheese. Welcome in. We're talking about genome evolution. Zoonomia project. Let's finish up our video on this as well. Lab manager wants me all nerdy. Me wants photos of me all nerdy and happy because she is also a Star Wars fan or a war.
And so we can compare our data with that data to understand how they relate and then predict what may make someone ill in the future.
Professor Kerstin Lindblad too started the project in 2015.
So there are very many common diseases such as schizophrenia or cancer where it's hard to find the elements in the genome that actually cause the disease. What we know is that most of these regions fall outside protein coding regions. So we have 1% of the genome giving proteins, but we have maybe 10% of the genome giving instructions of when a protein should be made in which tissue at which time point. So here we are trying to address the problem that we cannot find exactly which mutations cause disease. So by looking at the Zoonomia project data, we can see positions in the genome that are the same over 100 million years of evolution. They are probably functional and are therefore these regulatory elements that determine when proteins are made. We are looking.
So that's why looking at all these other genomes of animals will be beneficial to be able to glean insight into what exactly, hopefully, happening on the disease front with other animals as well. Her and her whole family are Star Wars fans like me. KK, did you see our new Star Wars animation? I thought you'd like this, KK. KK chat does not like this new Star Wars animation. But you hate it's okay, KK. You can hate the animation, KK. It's fine. It's fine, KK. It's fine.
Looking for positions that are very similar across species because they are likely functional. But we are also looking for positions that have changed a lot because those can be adaptations that make us uniquely human. For cancer, we can see that the mutations in those regulatory elements, not just in the protein coding sequence, can actually determine which genes run amok and cause the cancer.
Actually, these new findings are already being used in cancer research. Professor Karin Forsberg-Nilsson's research group is doing research on brain tumors that affect children. These brain tumors are malignant and are called medulloblastomas.
We are trying to find new ways to be able to treat the tumor better or maybe diagnose them better with the help of cancer genomics. It started with a genomic analysis of the 240 mammals to be able to pinpoint the positions in the genome that has been conserved over millions of years of evolution. If mutations in cancer occur in an evolutionary conserved position, we have reasons to believe that they are more functional for the tumor. Maybe these mutations, or at least a part of them, will make the tumor grow faster or potentially respond differently to a treatment. So that's why we have to go from the computers to the cell lab and see if the hypothesis that we have formed will actually be true when we test them on cells.
So she's just saying she actually has to test. We have to test our hypotheses from getting the data after getting the data and the potential information, rather than just saying, okay, this is how it goes, which is, so I could say it's just good science, but she's just going out and saying it's not a one-all answer. We definitely have to still test all of our hypotheses. Some of your video reactions put up that inform you what all happens. Thank you, KK. Ma'am, you have a blast, okay? KK, you have an amazing time in Star Wars land. And just so you know, I'm very jelly.
We culture medulloblastoma cells that come from patients with medulloblastoma, the brain tumor, and treat them in different ways to see if these mutations will give them an advantage to grow or maybe being able to resist treatment.
The researchers in the Zoonomia project hope that their unique dataset will prove useful both for understanding evolution and for understanding disease in humans and animals.
What's important is that this data is available not just for humans, it can be applied to any of our mammalian species. So when we use dogs to find disease genes, so dogs get cancer, humans get cancer, dogs get diabetes, human gets diabetes. And so we can use this research both to understand the canine genome and the human genome, and that's really important when we are trying to help understand disease both in dogs and humans.
Well, actually, it's available online, so anyone in the community can use it and intersect their clinical data with the information that we've found to predict what might be happening in their patients. It's a bit off yet to use in the clinic, but it's on its way there.
So it's pretty exciting there. Okay, I'm having a great time with closing guys. Thank you, Ariella. Ma'am, you sleep well. Have a lovely evening. And actually, we're rounding out our own evening now as well, Ariella. We are also rounding out our very own evening. So chat, that is 2 SciAnts News stories. There's a lot of similarities between the two. We were looking at both genomics on a human level and genomics on an animal level. And I hope you all enjoyed it and appreciated the very complicated nature of genomic studies and why they're important to study, as well as all the cool things that we can learn about from genomics. I hope it wasn't too overly dry. I hope you all enjoyed the science.